Welcome everyone. Today we're diving into predicting gene expression and splicing. So we are very fortunate to have three guest lecturers: Flynn Chen from Mark Gerstein's lab at Yale, Professor Xiaowei Xie from UC Irvine, and then Dr. Kyle Kai-Hao Farr from Illumina. So today we're going to be briefly introducing expression analysis, unsupervised learning, and clustering, just the most basic things that you can actually do with gene expression analysis. Then we're going to talk about the challenge of upsampling expression. How do you predict 20,000 genes from 1,000 genes, which is the first topic that Professor Xiaowei Xie is going to be covering today. And then a very cool take on this, which is compressive sensing. Instead of making single gene measurements, how do we make composite measurements that encompass multiple genes simultaneously? Then we're going to look at how do we predict gene expression from chromatin, and how do we predict reporter expression from chromatin features, which is what Flynn is going to be talking to us about. And we're going to then look at how do we predict splicing from sequence across thousands of features, which is also what Dr. Kyle Farr is going to be talking to us about. So let's dive right in. So up until now, we've talked about the landmarks of the genome that are gene regulatory, basically the elements that control the genes. And this is, a little out of order because the first thing that people think about when they think about genomes is RNA expression. And the way that we measure RNA expression has traditionally been using hybridization. Namely, you have a set of, let's say, 1,000 probes for every gene that you care about. And then you go and hybridize your RNA into those probes. How do you hybridize RNA? You basically first reverse transcribe it into complementary DNA or cDNA. And then you hybridize the cDNA against homologous sequence in each of your probes, effectively giving you a measurement for every gene in the genome. So these are the most basic building blocks. More recently, in the last 20 years or so, genome sequencing has become much, much cheaper than it used to be. So instead of first hybridizing your complementary DNA to an array and then measuring, you basically just simply measure directly. How? By effectively resequencing the transcriptome at every single iteration. So every single time you want to figure out which genes are expressed, you basically go through this wasteful effort of just resequencing the heck out of your genome. And this is only possible because genome sequencing costs have dramatically dropped by a factor of 7 orders of magnitude in the last 20 years. So these are the two technologies. And in the end, there's a lot of tricks and methods that you can do to call transcripts either de novo or to map your sequencing reads onto the known features of the DNA. And if you want to hear more about that, check out my fall course where we talk about RNA-seq analysis to great length. But for now, we'll just assume that we have the expression level for every gene, either through hybridization or much more frequently nowadays through RNA sequencing. What that gives you is effectively a matrix of activity. So you can take all your 20,000 measurements, put them all in one giant column of 20,000 genes for the human genome, and then you have a single vector of expression for every gene in the first experiment you did. And then you do the same thing for your second gene, and your second experiment and your third experiment, and so on and so forth, effectively giving you a matrix of which gene is expressed at what level in each of hundreds of conditions. And that's what gives us the expression profile of a gene. So that matrix can be seen either in the vertical direction or in the horizontal direction. In the vertical direction basically gives you a 20,000 long vector, one for every gene in the genome across that experiment of interest. For example, liver after cirrhosis, or brain after an infarction, or heart after cardiac arrest, and so on and so forth, or healthy heart, or young heart versus old heart, and so on and so forth, or this particular cell type in the heart that you have sorted. And next Thursday, we're going to talk about single cell RNA-seq where you just don't have to sort cells, but you just profile every cell. And there's a lot of deep learning methods associated with that as well. So, and that allows you to now start making comparisons across multiple dimensions. You can basically look at similarity of the expression vectors for a given gene across different conditions or tissues or cell types or experiments or age or gender and so on and so forth. Or you can look at vertically the similarity of different conditions to each other. For example, male versus female, young versus old, Alzheimer's versus control, and so on and so forth. Okay, so let's see who's with me so far. Great. So that's going to be the basic input matrices that we're going to be using for predicting gene expression. And then we're going to be complementing these with all of the matrices that we have already for the activity level of every regulatory region in the genome. 71, 24, 500. Lovely. Okay, so you can do all kinds of things. The most basic thing is don't bother with deep learning. The first thing you're going to do is just cluster. You basically just take all of your gene by condition, and then you cluster along this dimension to find similar conditions to each other. And then you just cluster them together. You're like, well, all of these experiments are very similar. It turns out that, I don't know, elderly people and people with Alzheimer's that are younger look similar. So maybe Alzheimer's is a component of aging or has some relationship with aging, and so on and so forth. So that's along this dimension, clustering, de novo clustering of different conditions. On the other dimension, you can basically say, well, it turns out that these 20 genes turn on and turn off in the same way across a bunch of different conditions and tissues. So maybe they have similar functions. And indeed, they're all involved in, I don't know, B cell and blood lineage and so on and so forth. Okay. So who's with me on the clustering part? So here you are in an unsupervised way, no training involved, just looking for conditions, experiments that are similar to each other, or genes that are similar to each other across the columns or across the rows. Okay, so let's do a quick poll here as to who's with me. Lovely. So again, this is the very basic building block, and this is completely unsupervised learning. So, so 67, 33, 0, 0, 0. On the other dimension, on the other side, you could basically look at classification. You could basically say, well, I already know a bunch of genes that are associated with, I don't know, T cell function or proliferation and so on and so forth. So instead of clustering the genes de novo, what I'm going to say is, can I look for additional genes that have proliferation-like signatures so that I can complete the annotation of some previously unannotated genes based on the similarity of expression. So this is the guilt by association. If you turn on and turn off with that gene, chances are, or with this group of genes, chances are that you probably play a similar function. Okay. So on one hand, we have clustering, which is completely unsupervised. On the other hand, we have classification. But again, what have we seen throughout these lectures? The fact that you can play games and sort of change things around and sort of build representations of your expression patterns using latent representations with perhaps sometimes dummy tasks. So I want you all to look at this matrix and say, oh, what could I do? I could, for example, erase this part and use unsupervised learning in a self-supervised framework to impute the missing data by sort of having massive training sets where I have taken out parts of my transcriptome, and then I try to re-predict them. And that will force the system to learn latent representations of your gene expression patterns and so on and so forth. Or you could basically say, well, hey, instead of looking at correlation, I'm going to use a deep learning approach to predict proliferation. So I'm going to use all kinds of additional nonlinearities and higher order features to learn about the proliferation class. Or I'm going to use a multitask learning to try to predict all of those classes jointly using this input. Okay. Who's sort of with me as to all of these really cool things that I can start now doing using the framework that we have already seen and the gene expression vectors combined perhaps with all of the chromatin vectors. So that's why we're having all these lectures, because you now have a set of tools. So 67, 29, 500, you now have a set of tools that you can use to go out and ask biological questions, learn representations of these systems. And again, I don't want you to think that deep learning is the only approach. You can use principal component analysis to basically look at what are the principal dimensions of variation, basically effectively shearing, rotating, scaling your space of gene expression patterns to learn the major dimensions of variation, and then perhaps projecting my vector of dimensionality from the gene vector to a meta gene vector, and from the condition vector to a meta condition vector. And that will basically tell you how perhaps every pathway acts and the membership probability of each gene in that pathway, and how major diseases function and the membership probability of each condition of each experiment in a disease condition. Okay, so that's dimensionality reduction, and you can do that with PCA. You can also do low rank approximations of your matrices using these singular value decomposition approach of keeping only a subset of your singular values and then zeroing out the remaining ones, which effectively gives you a provably optimal lower rank approximation, assuming a particular distribution of your model. And then applying PCA on the MNIST digits gives you this. And then there are other approaches. For example, t-SNE gives you a different type of embedding of a two-dimensional space that allows you to represent these classes as much more separable from each other by effectively learning a lower dimensional space that maximally separates the features from each other. And we're going to talk a lot more about t-SNE at the single cell lecture next week. And you can also use autoencoder. You can basically say, I can compress my expression matrix to a lower dimensional representation, and I can then expand it back out to a higher dimensional representation by effectively using self-supervised learning through a bottleneck of forcing a lower dimensional representation, the same way that we saw for images earlier. And this is some of the key ideas that we're going to be talking about today. We're going to be talking about upsampling. For example, how do you predict 20,000 genes from a subset of genes? So in deep learning for image analysis, we've basically seen how you can, in fact, learn representations of the world in high dimension and then use self-supervised training to basically take higher resolution images, subsample them, and then use this as the X and that as the Y, and then predict the upsampled image, effectively learning how to upsample images, and also as a byproduct, learning representations of the world. And you can do the same thing with subsets of measurements in genomic space. You can build these deep learning architectures for upsampling images by upsampling at different places in this image. And then you're going to hear about one such architecture from Xiaowei Xie, who's going to be giving the second guest lecture today. You can also think about sort of a very different way of doing your measurements. Instead of just measuring a subset of genes, you can actually build composite measurements using combinations of probes that together capture linear combinations of your gene expression. And that's known as compressive sensing. And you can also ask about how well can I predict gene expression from chromatin information. If you look here at the genes that are expressed at different levels, what you see is that their level of DNA methylation varies greatly. And therefore, if I have DNA methylation for these genes, maybe I don't need to measure gene expression. Maybe I can just predict gene expression based on the pattern of methylation that I see across my sequence. And that's very exciting because for some samples, RNA degrades very rapidly, but I can find DNA methylation from much less well-preserved samples, and then maybe predict the expression of genes corresponding to that. And you can see the same kind of relationship with histomodification marks. If you look at genes at different quadrants of expression, the most highly expressed genes have much more H3K27 acetylation, and the much more lowly expressed genes have much more quiescent state in their gene body regions. So you can perhaps use chromatin information to start predicting gene expression levels, which is going to be the first guest lecture that we're going to hear today. And then Deep Chrome, for example, is one approach for doing this by combining multiple features at different distances in order to sort of make that prediction systematically. And then Attentive Chrome uses attention mechanisms similar to what we talked about for the transformer model when we talked about recurrent neural networks. Again, I want you guys to start thinking broadly about all this. So again, we're going to have a guest lecture by Xiaowei on how do we predict expression and chromatin from these features. And then you will also hear about how to predict reporter expression from chromatin. And then this concept is that you basically can build thousands of reporter constructs simultaneously, test them in very high throughput fashion, and then effectively infer which gene regulatory regions, which test fragments, which candidate gene regulatory regions actually drive gene expression. And you can now go back to the genome and say, okay, what are the coordinates of these fragments? Can I now use a machine learning approach or a deep learning approach to use the chromatin features in the primary sequence to predict whether these fragments are going to be driving expression? And you will hear more about that from Flynn Chen, who's a member of Mark Gerstein's lab at Yale. And then the third guest lecture today is going to be about predicting splicing directly from sequence. The idea is the following. Every single cell expresses not just a contiguous block of RNA, but spliced RNA, which basically mixes and matches exons for different tissues and cell types. So you can think about the inclusion or exclusion of every exon, and then use that in either a Bayesian neural network or a representation neural network or a deep learning neural network to basically start predicting the inclusion or exclusion of individual exons and then the specific features in that sequence where these are happening. And we're going to have Kyle Farr from Illumina, who was actually previously at Harvard, MIT, and at the Broad, talk to us about deep learning for splicing prediction. All right, without further ado, I would like to ask if Flynn is there. So Flynn, take it away.
Sounds good. And thank you, Professor Achilles, for inviting me to come talk to you guys today. My name is Flynn, and I'm a graduate student at the Gerstein Lab here at Yale. I'm super excited to talk to you about some of the work that we've been doing on using deep learning to predict enhancers specifically in the human genome. And the title of my presentation today is called Where Before What, a weakly supervised framework for precise enhancer localization. And with this title, I sort of want to give you a little bit of intuition on what is weakly supervised learning. And my goal for the presentation today is to sort of present to you how we operationalize the task of enhancer discovery into this weakly supervised framework. So here is the link to our most recent manuscript if you are interested. So to give you a little bit of introduction, enhancers are regulatory elements that increase the transcription of a gene. And it's very important to map out these elements because there are potential genetic drivers to diseases. And earliest methods have focused on unsupervised approaches like Chrome HMM because of the lack of supervised data. But with recent advances in experimental techniques like Professor Achilles had mentioned, the MPRAs, we can massively probe enhancers and obtain a large labeled data set, and we can start switching from this unsupervised paradigm into a supervised paradigm. And so specifically, there is a type of MPRA, massively parallel reporter assays called StarSeq. And what this assay does is for a cell line of interest, for example, if you have the genome of that cell line and you fractionate that into 500 base pairs of fragments, and you insert that in front of the luciferase gene in this plasmid vector, and you transfect that vector into back into the cell line of interest. And if that fragment is an enhancer, then you would see correspondingly an increase in transcription of this luciferase gene, which is detected by mRNA expression. And so using this method, you can probe for every single fragment in the human genome, whether if it's likely to be an enhancer or not, using a very high throughput fashion. But there are some shortcomings to the experiment. For example, each of these fragments are evaluated outside of its original epigenetic context. What that means is that the ability for the
Flynn, we seem to have lost you. Oops, my guess is connection problems. I do have these slides, so why don't I start sharing, and then hopefully he'll reappear. So the key concept here is that we can basically systematically measure the reporter activity for every segment in the genome, and then ask, based on how much expression every segment drives, can we now start predicting the amount of expression it will drive based on the corresponding chromatin features that were at that segment at the time. So the way that Flynn set this up is by looking at 5 different cell types where StarSeq, this reporter experiment, was available, and then ask for each of them, can I predict the StarSeq result using a set of features that we've already talked about in the class? And these features are DNA accessibility, measured through a tag-seq or DNAs, histomodification patterns using chromatin immunoprecipitation with sequencing, which include acetylation of K27, of lysine 27, of histone 3, K4 trimethylation. These are marks of enhancers, of promoters, of enhancers, and of promoters. And not exactly, because not exclusively, because there's also a lot of overlap. So his idea is, can we now use all of these as features to predict StarSeq expression? The way that they set this up is by constructing this input matrix of all of these different marks across thousands of locations in the genome, one for every gene, and then asking what are the nearby features, the nearby chromatin features that allow us to now start predicting this systematically. And the output level was simply whether something was found to be indeed active as an enhancer in the StarSeq experiment, or whether it was not found. So it's a binary classifier in the end. So that's the first part, and that's what everybody would have done. But what's really cool and neat here is that they then said, can I now, from these intermediate representations, go back and start predicting where in the genome sequence is that particular element? And Flynn, whenever you're back, take over.
Yeah, I think my internet is a little bit unstable when I'm sharing and having this network connection all the time.
Why don't I give you control of my slides, and then you can control my pointers instead.
Yeah, perfect. That's awesome. Yeah. Let's see. Yep, I think. See. Yeah. So basically, as Professor said, that we have this convolutional neural network that could predict whether, if there are, let's see. Yeah, so we can predict with a binary classification of whether if there are enhancers in this region or not. But with a positive prediction, what we, in addition, we can do is we can take that the gradient of the positive forward pass and use this specific method called GradCam, which is called gradient weighted class activation mapping. And we can backpropagate that gradient to specific layers in our network that generates these feature justifications, as well as basically an importance score highlighting which area in the input is most important in our in making the decision for our model. And we can take that subset of that input and redefine that as basically a core enhancer region that is much more higher resolution and compact compared to the original input, which is a 4 kilobyte window.
For everybody on the Zoom call here, I really want you to think about not just this particular example, but this as a more general principle of basically going back and fitting particular curves in these contiguous signals to then have a higher order representation. For example, if you're thinking, where is the cat in the picture? You could just say, oh, what lit up as a cat, or you can take a shape of a cat and then project it back and then ask, where is that lighting up? So that's what Flynn is doing here. He's basically going back and reinterpreting this by taking various shapes and then projecting them back through that convolutional network. Continue, Flynn.
Right. Let's see if I can advance to the next slide. Let's see.
There's a little bit of a delay in your network, so don't scroll multiple times.
Okay,
Just tell me when to scroll, and then I will scroll.
Okay. All right. Are we on slide 6? Let's see.
Yes, we're on slide 6.
Okay, cool. All right. So yes. So as you can see, when we pass on the original image into through a convolutional filter, it generates an activation map. And each convolutional filter learns the most important features in making an active prediction. And therefore, the activation map produced by that convolutional map will highlight that particular feature that was trained. And a linear combination of these activation maps by multiple convolutional filters in a particular layer actually produces a heat map of what you would see in image A here, image I, that highlights basically where the dog is in the image. And essentially, this is the idea of weakly supervised learning, where we did not provide any location information to this model. Unlike object or semantic segmentation, we only had a label of whether if it contained a dog or not, and we are able to infer that location information. And we aim to do that for enhancers as well, where we only have coarse annotations of whether if enhancers exist, and we want to figure out the precise location of where enhancers are using the same method here by doing a second pass. And that's the intuition behind our object detection method. Let's see.
So you're on slide 7 now.
Yeah, let's see. Yeah, so this is the result for our cross cell line and cross chromosome validation. We show that given the input features, the chromatin accessibility and the histone marks, we're able to make accurate predictions on whether if this is a star-seq enhancer. By cross cell line validation, we show that we can generalize the new cell lines, and cross chromosome validation indicate that we can generalize the new genomic loci. And here are some of the example predictions. In the positive predictions, you see that there is a lot of epigenetic activity going on, and in the negative regions, there is almost no activity. And so we benchmarked our model with the state-of-the-art matched filter model that uses a shape matching filter and a linear SVM for classification on this ENCODE enhancer challenge dataset. And we show that we're able to outperform the previous state-of-the-art model in almost every tissue type, with some even with 15 to 20% margin. And we performed a case study in neuroprogenitor cells, hoping to discover neural-specific enhancers. And we see the feature justifications here that we focus on DNase and 3K27AC and 3K4ME3, these strong active histone marks, and these marks interact and form this higher level feature on the right. And we're able to use this higher level feature to basically say, we're gonna shave off these regions that are not relevant when our model is making a prediction, and obtain a refined and higher resolution set. So here we have the 4 KB window original prediction set and the refined set. And you can see in our statistics here that our refined set has 13% of the coverage as the original set, but account for a higher proportion of transcriptional start sites. Through phylogenetic conservation analysis, we show that the refined set is also more conserved in 100 different species. And we also evaluated intraspecies conservation with the rare SNPs, because if a certain region is more conserved in intraspecies, it will have more rare SNPs due to negative selection. And we also show that our refined regions through our GradCam method is more enriched in intraspecies wise.
So we really have to run to Xiaoyi's talk. So thank you so much for a wonderful presentation. Everybody, please give Flynn big thank you.
Yeah, thank you.
Xiaoyi, I see that you're already here. Do you want to share your screen?
Hey, good morning, everyone.
Hey, good morning. So Xiaoyi is a professor at UC Irvine, and he's also an alum from MIT on the Broad. So we're very fortunate to have him back, and someone that has truly done so much work in deep learning, machine learning, and just general AI research. So Xiaowei, take it away.
Thank you. Good morning. Not good morning. I kind of get it.
Good morning in California.
Good afternoon, everyone. Thank you, Manoli, for inviting me to be here. Let me share the screen first. Let's see.
That's the beauty of Zoom, the fact that you can have friends from all over the world join.
That's right. So that's actually very convenient. Let me see if I set Windows. Can you see my screen now?
It works very well. And we can also see your pointer, so you can guide us through the slides as well.
You can see my pointer?
Yeah.
Oh, cool. Wonderful. Great. Yeah. So thank you, Manoli, for inviting me to be here. So it has been a long time. I have not been back to the MIT for quite many years. So that's right. When Manoli invited me to be here, I said, I might jump on. I said, I'd be glad to come here to share some of the work we have been doing in this area with you guys. So it's exciting. So today, I'm going to talk primarily on the deep learning applications on the gene expression analysis.
So Xiaowei, for some reason, we only see your original screen. We don't see the projection. So I don't know if you shared just one application rather than sharing your screen.
Oh, let me see. Let's see. So you only see.
Yeah, so share your whole screen rather than just the application, because when you project, it effectively becomes a new application.
Okay, share the entire screen. Okay, let me see.
Exactly.
So I think the reason, because when I do, where is it?
Wonderful.
All right, is this better?
Yes, now it's better.
Okay, all right. So this is the full screen here. Okay, yeah. So let's go back here. So today I'm going to talk about the applications of the deep learning on the primarily on the gene expression part of that. So before I talk about that, of course, this course is dedicated on applying deep learning to the to biology and medicine, primarily genomics. And as you already learned from Manoli, is that we have lots of this biological data emerged recently due to high throughput sequencing technologies of that. I believe you already talked about the RNA-seq, and then you can talk about the ATAC-seq, and also single-cell genomics as well. So as you can see, what you have here is a very large scale, this type of genomic data sets. So of course, biology, you need time method here to kind of visualize, kind of integrate, interpret this type of data sets for biological discoveries. So certainly, however, a lot of challenges in terms of analyzing this type of data set. There's have high dimensional, many times it's very noisy. And how do you kind of extract signals from those high dimensional noisy data sets here? It's always a challenge for many of you, for us as well, in terms of developing methodology, how to do that. And especially for the single cell genomics data set, there are a lot of this kind of called a sparse or missing data sets kind of hidden within this type of technology. So this particularly kind of important for developing methodology that taking those type of factors into account. So the research in my lab here, over the years, I started with my career with the work with the Manolis when I was at the Brody Institute many years ago. Then since I've been a faculty at the UC Irvine here, so we have been still continue to develop methodology for studying kind of the field of the genomics, so I'm particularly interested in how to develop method try to combine kind of multi-modality genomics data sets. As you heard from Manolis, learned from Manolis, that nowadays we have so many different types of techniques and high throughput methodology have been developed over the years. And so the ones, to be honest with you, the one I'm most familiar with is kind of RNA-seq, ATAC-seq, DNA-seq, but now there are so many newer type technology, I don't even be able to keep track of those methodology. I kind of listed some of them here. So certainly it's going to be very useful. How do we develop methodology that able to combine those type of multi-modality data sets to allow us to look at the underlying biology from the combinations of data sets. So that's something I kind of keep a keen interest in this direction of that. So of course, there are also recent methodology people have been proposing in the machine learning field that combine kind of multimodality data, try to understand the system better. So one of the kind of the hallmark application is how to combine kind of visual signals, computer vision with the natural language processing, and hopefully combine them together can help us learning the systems better. So this is a kind of interesting research area as well, just in the field of the machine learning. So over the years, my lab at the UC Irvine here, we have developed a few tools for the using primarily deep learning methodology for study, for analyzing genomics data sets here. So I listed a few of them here. So I will not be able to go over all of them. I just maybe I just spend 1 minute to quickly go over each one of the methodology, what they are doing, and if you are interested, and of course you're welcome to ask me in this talk here, or just go directly to some of the GitHub here and check them out. So the first tool is this called DanQ. So this is a deep learning methodology for kind of quantifying the function of the DNA sequences. I'm sure you learned from the Manoli that human genome, only small fractions of them are actually coded. The majority of them are non-coding sequences. So over the years, people are very interested in identifying the functions of the so-called non-coding sequences. We have experimental techniques in terms of read out signals from those regions, but in general, it's pretty hard to pinpoint down the function at the individual base levels. So this is also relevant for the disease studies, because many of these genetic variants are discovered within those non-coding regions. So and people want to know what potential function, what type of variants are functional, what type of function may not be functional. And then, so this is a tool was developed, try to kind of give some ideas in terms of functionality of the individual variants and whether they are functional or not, what can we say about them? So that's a tool called DanQ. It was developed quite early stage of that. And the next tool is called a GEX. So this is a tool that we developed for gene expression analysis. So I'm going to spend time to talk about this one today because as Manoli is asking me to talk about this one. So this one here, we developed neural nets here to try to predict gene expressions. And so that was developed so that we're going to spend some time to talk about this ones here.
Erwin, you can go really fast on DGEX and then spend more time on Sailor. That's fine.
That's okay. And then the third one is a factor net. So this is the one we're trying to predict kind of a gene transcription factor binding in the genome-wide fashions. But over the years, people have done a lot of this type of so-called called ChIP-seq analysis, and those analyses is able to identify transcription factor binding in a genome-wide fashion through experimental procedures. And we're interested in utilizing those type of large-scale data sets, whether we can come out with a model that can predict kind of transcription factor binding in a cell type specific fashions. And I'm sure you heard about that because this type of methodology is one of the kind of early applications of the deep learning in genomics. So the idea is that previously we modeled the DNA sequence primarily through this so-called position weight matrices. And people realize that we can also use these convolutional operations to model the DNA bindings. It seems like pretty effective. So this is the work here is work along this type of lines then. And I'm sure if you have not heard about that, I'm sure Manoli will talk about this part. So then the next part of work is called SC-FIN, and this is kind of extensions of the factor net. So in which we try to come up with a model that can predict transcription factor binding in single cells, because in single cells it's very hard to kind of carry out those type of chip-seq experiments because of limitation of the material so far. And but however, people are interested in whether we can have understanding of individual transcription factor binding in single cells. So we developed some methodology for that purpose. And then the next one is called a U-fold. And this is the one for the RNA structure, secondary structure predictions. So and we found that applying some of the techniques can be pretty effective in terms of predicting the secondary structure of the RNAs as well. So the next final one I will talk about in today's lecture is called the Sailor. So this is a methodology we just recently developed for utilizing deep generative models for studying this single cell ataxic data sets, and primarily focus on the so-called the ideas of the invariant representation learning. So like to learn hidden representation that is kind of reflect the underlying biological properties of the cells instead of the experimental artifacts. So we're going to spend time talk about this methodology here. So of course, all the tools we develop are open sourced and available in the GitHub here. If you want, you can check out. All right. So today's agenda here. So first, I'm going to talk about this DGX, this models here. So this one we're going to use deep neural net to predict gene expression. So it's pretty straightforward applications of the deep neural net. I should be able to go over that pretty quickly. And the next one, we're going to give a little bit overview on the deep generative models for the genomic side of that, especially focus on this variational autoencoder types model, VAE model. So you learned that from Manolis a few lectures ago. And here you'll see how people apply the VAEs to study genomics, and in particular for the RNA expression data sets of that. And then the next I will do kind of talk about one extensions of the deep generative models for the ataxic analysis. In this case here, we think about how do we remove the kind of experimental confounding factors from the data set and allow us to learn a invariant representation, like representation reflect the biological properties instead of experimental artifacts. And this is involves some of the new ideas relate to the deep generative model. I'd like to introduce this part in the third part of that. And finally, just to probably quickly mention what will be the idea, how do we combine this multi-modality data set, come up with a model to learn a representation that can share across different types of data sets. So that will be the last part. Hopefully I can go over them in today's lectures here.
We only have 12 minutes left, so I would really speed up the DGX part.
Okay, so I'll keep my clock here and make sure I finish on time here. Okay, so the next one is, so first we'll talk about DGX. It's pretty straightforward, and I will go that pretty quickly. I'm sure all of you understand this part here. So this is the part was a work done a few years ago. And then the main idea is actually collaboration with a few folks here at the Broad. So especially Arun Subramanian, I'm familiar with that. He is still at the Broad here. So this is related to a project that was done quite many years ago when I was at the Broad, way back in 2006 and 2007. So the idea was originally called a connectivity map project. So people realize that we can quantify the profiles of the cells in terms of, based on the perturbations, There are many different types of perturbations, right? Perturbation can be reflected in different cellular types, or can be reflected if you apply drugs to the cells and the cellular state will change. So there are many different types of agents you can apply to the cells, and then the cell state will change. So the primary goal of the connectivity map project is try to provide a comprehensive characterizations of the cellular state in terms of the expression values, right? So of course we can characterize cellular state in terms of epigenetic modifications as well. But initially the CMAP project is trying to characterize state in terms of the RNA expression values of that. So in 2006, and then they provide this pilot in a data set, they're able to profile the expressions of genes in the cell lines through many different perturbations, primarily through some small molecules, some drugs, and so on. So that was a very large scale data set at that time. And the idea is the author originally has that maybe you can provide type of a search engine type of idea. Once you have this data set in place, and what you can do that once you have a profile, you've done some G expression analysis, you can use them as a query, and then you can do a pattern match across this database. Well, immediately tells you how this particular perturbation is related to the ones in the database. So that immediately provides the link, potential link here between your small molecules and maybe the RNA expression perturbation due to disease, due to other agent perturbations. So build this type of links between those type of profiles. So that would be very helpful for understanding both biological process, even for drug discovery through this type of methodology. So that was the original idea. And then people are very excited by this. They want to kind of extend from this relatively kind of pilot small scale data set to much larger ones. And however, in order to do a larger ones, and they will think about how to scale up, of course, and the cost is a concern. And then that time they propose this idea is that maybe we can generate a profile of the genes, not across all genes, maybe based on small subset of genes. And by doing that, we can generate profiles quickly at a much cheaper cost. And so that's the original ideas of that. So they propose a technology platform. And based on this 1,000 gene solution, in this case, we only profile 1,000 genes instead of 20,000 genes across the entire genome. And so the idea is that because the expression of the genes are highly correlated with each other, and it might be that by profiling this 1,000 genes, you will be able to infer the remaining, all rest of the genes. So that's the main ideas behind that. So how do you know this is true? Because you can take out the existing gene expressions and do a simple dimension reduction. And you see how many kind of a principal component that can be used to explain the variance you observe within the data sets. At that time, it was very surprising. And they're now saying that probably with only 100 to 200 genes, that can explain 80 to 90% of the total variance observed in those entire data sets here. So that was shocking because it's saying that you only have this few hundreds of factors can explain the entire universe of gene expression. So that I was surprised by that. But this motivated them for developing this methodology. They pick up those those 1,000 so-called landmark genes, and they use this the technology platform called Luminex Beat, and then they can profile the expression, and then they can. So by doing that, they can kind of doing this in a very high throughput way, can generate profiles with millions of samples. So that's how they did. I believe the project was still ongoing. Later on was evolved in something called a links program. And then they are able to generate millions of profiles with those 1,000 genes. I believe they're still expanding that. So it's very large useful research if you want to look at this part of that. So however, and what we initially, when we worked on the problem, what we want to know is this, and we're interested in, so okay, now we have this landmark genes, small number of them, only 978 of them. So the question whether we can infer the remaining 20,000 genes using some computational techniques, right? So that's the purpose of that. And we know that we should be able to, based on this type of simple dimension reduction analysis, that we should be able to do it reasonably well. And however we want to do it kind of accurately, improve the precision, the accuracy of that. So at that time, so the neural nets is not very widely used in this type of in genomics. And then we started this approach to do that. And it's pretty straightforward, right? So what we do here, we input here will be kind of 978 dimensional vectors here, go through several layers, multi-layer perceptrons, feed-forward neural nets, and we're going to predict those 20,000 target genes in a multitask fashion jointly. And then we train them through backpropagation. So that's kind of pretty straightforward idea. So at that time, it's harder to implement because there's no PyTorch, no TensorFlow as you do convenient today. So that, and the question is, how do we train this model? So that we train these models here with those data from the GEO. So GEO data set does contain the expression profile of the entire collection of the genes. So either a matrix array in the old time, and then later on, most of the data came from the RNA-seq data type that. So we train these models with this collections of the GEO data set, and then we say how well we can do based on this roughly 1,000 landmark genes predict remaining genes here. And it turned out it's doing pretty well. So at that time, the baseline performance is based on linear regression. So it's kind of very easy benchmark to beat. And by utilizing those type of deep neural nets, we can do much better than those simple kind of linear regression models here. So this is a kind of a overall error rate here. You can see linear regression as here. Now, if you change different type of architecture of the neural nets with how many, by choosing different hidden units here, and how many layers of hidden units, and you can see that the model does improve when the systems become more complex. It can fit the model better. So
we're really short on time, so I really wanted to have enough time for the other parts. So don't.
Okay, let me. I think I'm okay. I'm done with this part here. So I'm pretty quickly here. So this is the basic idea. I think it's pretty straightforward. I think and then that's the initial application of this part here. So I will skip this part here. So next I'm going to talk about this part here, and then later on people think about how to utilize this deep generative models for genomics of that. So that this is more or less related to the variation autoencoder model you talk about. So because the first methodology I talk about here is kind of a very rare application of the gene expression analysis based on the neural net. However, most of the time, as you know, for the genomics data set, we do not have labels, right? Most of the time, we only have the input X without the Y. So in this case here, this type of unsupervised learning is sometimes more relevant for the genomics data sets of that. So for these reasons here, this deep generative models has been kind of widely used for studying genomics here. So I think it's related to this underlying this so-called manifold hypothesis. So the idea is that, so we have this kind of high dimensional data set. It's pretty, and very complex, highly noisy. And most of the time it's difficult to identify underlying patterns by direct look at those high dimensional data sets. And maybe if we can able to map those high dimensional data sets here into a kind of low dimensional embedding or manifold of that, and then we study those signals within the low dimensional embeddings of that, that might be more helpful for us to understand those biological data sets. So that's kind of main ideas of that. And the question is, how do we do this? First, we have a lot of this kind of a traditional dimension reduction methodology. And later on within the field of the deep learning, so the method primarily is based on these ideas of the autoencoder idea. So that I'm sure you learned that from Manolis. So the main idea is this: you have your data sets here, X here, you input here, go through layers of the computation, and then we represent them using a latent representation here. And then this latent representation later on, so this most of the time is called an encoding part of the models. And then we can, once we have identified our representations of the input here, maybe we can recover those input here through this latent representation. And this is type of a kind of traditional autoencoder types of models here. And then you can train this autoencoder by mapping by make sure that the output here match the input here. So that's a traditional autoencoder type model. And however, this autoencoder have a few issues here, right? So there's no constraints on the representations of this latent representation. That can be any type of distributions. So people realize a lot of issues there, and then this type of model tend to susceptible to overfitting, a few other issues. And also, you cannot generate samples based on those traditional autoencoder types of model. So that is the reason people propose this type of those type of ideas based on those deep generative models. So this is the kind of deep generative model has a long tradition in the field of the machine learning. And so the idea that how do we model data through type of probabilistic models? So in the case of the latent representation, if we believe that those data sets here have a simpler kind of latent representation, we may write down that probability distribution relatively easy through a generative models here. So in this case here, we're going to assume those latent variables are Z variables here. So these are, suppose you have data, give input data X here, and the true underlying representation is through kind of less complex, lower dimensional Z variables here. So that's the latent variables here. We want to model the distributions of X given Z with certain parameters of that. And then we're going to assign some prior to the distributions of Z here. And in the end, model, by doing that, we can kind of marginalize over Z to obtain the marginal distributions of the X So that's kind of a very generic framework for the latent representation of generative models here. So in this case here, you have some parameter theta here, tells you how do you model the conditional distributions of x given z. And then we can learn those parameters based on the given data sets here. And then most of the time we learn that by construct a loss function based on the log likelihood, or particular negative log likelihood here, and take a log and then try to optimize parameter of that. So however, this is kind of very generic framework here. And however, people realize it's kind of very difficult to learn those theta here based on this model, because in order to calculate the marginal distribution, you have to do these integrations here, marginalize over the entire distributions of z. So that most of the time is intractable. It's very hard to do this part. So that is the reason people propose some of their, most of the time proposes an approximate inference methodology to solve this problem. So the most popular one is based on this called a variational inference. So in this case, so we're going to propose some auxiliary distribution on the distributions of z given the x here. And that was modeled through this Q distribution here. So this model the distribution of z given the x here through some parametric forms here. And then we're going to try to, you can show that this log likelihood here is bounded by this lower bound here. So this lower bound is interesting. You learn from monodics, right? So this is the first term is this data term. How likely is the observation, the log likelihood of the data? But instead of marginalize over the margin distribution z, you now marginalize over the kind of proposed distribution z given this x here. So this is your proposals of that. That's called auxiliary distribution. And then at the same time, you want to minimize the KL distance between those distributions and your prior distribution. So the idea that you want to ensure that your posterior distributions here is close to your prior distribution, sufficiently close. Of course, there's always the balance between these two terms of that. When you try to maximize this, you want to minimize the KL divergence, which means that you want to make sure that your proposed distribution is similar to the prior distribution. And at the same time, you want to make sure that you have sufficient power to model the distributions of the observed data sets here. And so that's the general ideas of kind of overall generative models. So in the field in the field of this called a variational autoencoder, what the people propose that I can model both this conditional probability p theta x given z and also the auxiliary distribution through neural nets. So that's the main idea behind that. So that came out with this ideas of so-called encoder part and also decoder part. So decoder part is simply model the probability distributions of x given the latent representation here. So this part of that. And the encoder part try to propose kind of a good proximate distribution of the latent representation given the x part of that. So so it's parameterized by parameters, the theta and the phi here will be the weights of the neural nets in this framework. And in the end, we can train them just simply by minimizing those this variation of the negative log likelihood and minimize them. And that turns out to be very efficient. You can train them very quickly through this sort of traditional deep learning training framework. Was proposed in 2013 by Kima and Wellens. So however, in general, this framework still have some issues, in particular because calculate those expectations is still not easy. And then one of the main ideas that you can use so-called a reparameterization trick here in such a way that when you sample those latent representation, it's easier to calculate. So the main idea that in this case here is particularly if you assume your prior on this latent representation is a Gaussian prior. If you make that assumption, so Gaussian prior is a very strong prior saying that you have this, suppose you have k latent variables, you assume they are independent of each other. Each one of them follows the simple normal distribution. So in that case, you can kind of sample those distributions of z here is very easily through by just learning the mean and the variance of this conditional distribution. Instead of learning entire distribution, you say, I just learning the mean and and the covariance of this latent representation. And after that, you can sample the areas.
We have another guest lecturer, Kyle, who has been patiently waiting. And unfortunately, we're really out of time. So I don't know if you can do something about this.
Okay, so what time do I have? What's the?
14 minutes ago, I told you that you had 12 minutes left, which was already 6 minutes over what we had planned.
Oh, sorry about that. So I thought I was 11:30. No. Sorry, I thought I have time to 2:30.
No, when I told you 12 minutes, it was 14 minutes ago. I'm sorry to be so difficult. It's just that we have another guest lecturer who's waiting in line.
Okay, sorry, give me 5 minutes. Let me wrap up. Is that okay?
Okay, 5 minutes.
Yeah, 5 minutes, yeah. So this is overall this variation autoencoder model. Of course, you learned from Manolis. I'll skip that part of that. So people apply those kind of variation autoencoder models here to the RNA expression analysis of that. So there over the years, the many models have been proposed here. So what are the main ideas here? So the main idea is that we like to capture because we are modeling the generative model for the RNA-seq data sets here. So the main idea is that how do we kind of capture the characteristics of the RNA-seq data sets? So most of the time, because RNA-seq data set is kind of a discrete, is kind of a quantitative, is discrete, is not a continuous numbers of that. So that in this field here, people typically use something called a zero inflated negative binomial distribution to model the distribution read count of the RNA-seq data sets of that. So you can think about this kind of how do you produce integers based on probability distribution of that. So that has been very useful. So this is kind of a model here, initially proposed in DCA framework here. And so main idea is that you still use autoencoder here, but then use the final part is not based on Gaussian models, but based on the zero inflated negative binomial. So I'll skip that. There are a few parameters to do that. So that's kind of main application. Most of the deep generative model is applying those type of variational autoencoder combined with the zero inflated negative binomial models, and then you can learn that from the in end-to-end fashion. So there's this work here called SCVI. Manoli mentioned that the authors of the work will come here and talk about it. So you'll get an opportunity to learn more about this hour here. So then I will quickly go over the last part of that couple of minutes. So the idea, many of the ideas about this type of deep generative model is that we learn this latent representations. So let me see. So we have given the input here. So we learn this latent representations here. And however, we realize that those latent representations here most of the time can reflect a lot of those experimental artifacts through which we generate these data sets here. So in particular, for many of the genomic data sets here, so there are two major confounding factors here. One is so-called batch effect. So in how the experiments were done in different patches of that. And also in terms of in this RNA-seq or RNA or ATAC data sets here in terms of coverage of the reads. And that can have a significant impact on the representation of Z if you do not count for them. And then you look at this latent representation, you will see that latent representation are primarily impacted by those type of confounding factors. So in this field here, so we are interested in how do we remove the confounding factors such that we learn the representation only reflect the underlying biological properties of the cells instead of those experimental artifacts of that. And then we came up with this idea that maybe we can learn those deep generative models here through a conditional generative model. So the idea that we not only generate data sets here based on the underrepresentations here, but also based on those conditional, those underlying, those confounding factors. Sometimes we know what are the batch effect, what are the coverage of that. And what we also want to know is I want to make sure that, so those Z and C's here are kind of independent of each other. So once I have C here, I'll make sure this latent representation is completely independent of C. And that's how we came up with the framework here to learning those latent representation, in which case here, what do we do here? We do the deep generative models here, but then we'll make sure that we want to minimize the so-called mutual information between the Z and those latent representations here. And so overall loss function is slightly different. Not only want to make sure we'll be able to generate data well, but also make sure that the mutual information between them is minimized.
This is a beautiful, beautiful idea, Xiaowei. Wow, very cool.
So, and then I think I will wrap up here. So then you have, and we have some work, and then if you would like to more, and then we'll be able to see more of that. But I think I will wrap it here in the interest of time. Okay.
Thank you so much for a very
Thank you guys. Yeah.
talk. Kyle, can you please share your screen and get going?
Hi, can you guys hear me?
So big thank you to Xiaowei. Yes, we can hear you, Kyle. This is great.
Okay, great. All right, so I have about 15 minutes. I'll try to take you guys through our work on splice prediction. You guys can see my screen?
Yep, very nice.
Okay, great. Yep. All right, so I'm the principal investigator at the AI lab at Illumina. And our goal over the next decade is to try to understand every possible variant in the human genome. And this is really a very daunting goal. Right now, 99% of the genome is non-coding. Most of that we don't understand how the variants in there work at all. Even for the 1% that's protein coding, 99.9% of those are variants of unknown significance. Only 0.1% are present in databases like ClinVar. So Illumina's interest in making the genome much more interpretable is directly related to our business. We're very interested in making genome sequencing useful not just for a small fraction of the population, but for everybody. So our hope is that as we make the genome more interpretable, more and more of the population will get sequenced as soon as possible. We're also very interested in interpreting non-coding variation in general. As you know, right now, most clinical sequencing is performed on panels or exome sequencing, and that completely skips the non-coding genome. If we're able to bring in pathogenic mutations and provide people with the tools to interpret them, then you can imagine that we can move these markets to whole genome sequencing, which would be a great thing for Illumina. So this is why we're investing very hard in this area. And this is SpliceAI, sort of our first demonstration of the power of deep learning networks at interpreting these non-coding variants. So if you think about where the diagnostic yield for rare disease might be, if you just look at sort of what are the most highly conserved nucleotides in the genome, so I'm not talking about all conservation, but just the ones that are very deeply conserved across vertebrates, what you see is that most of the really deeply conserved sequence is actually protein coding. This is not true for a less conserved sequence, but then beyond the protein coding sequence, you can see that it's kind of divided among UTRs, promoters, enhancers, and nearby splice regions to exons. So we've essentially been investigating each of these areas with deep learning to identify what are the sequence to function models for interpreting variation. So, of course, the challenge for finding pathogenic mutations, especially in the non-coding genome, is very challenging. A problem looks like this. So it's essentially a problem which, unlike vision or language, humans have no innate ability to do. So we believe that pretty much in the future, all interpretation is going to have to go through AI and deep learning. And then just for a primer on splicing itself, if you guys recall from Molecular Biology 101, in higher organisms and vertebrates, exons and exons have to be spliced out of the original gene body. So typically the gene body is 10 to 100 times larger than the actual protein coding sequence. And splicing is the process by which most of this excess genetic sequence is removed. And the primary nucleotides are the GT and the AG at the beginning and end of the intron. They're called the splice donor and splice acceptor. And those are sort of the critical elements for defining the intron. But as you can see, these are just like 4 nucleotides of specificity. So it's clearly not enough to find exon-intron boundaries with nucleotide level precision in a typical gene body, which is usually hundreds to megabases of sequence. So introducing SpliceAI here. So this is a deep convolutional neural network that takes in about 10 kb of input sequence. And Basically, for the entire gene body, it's a sequence-to-sequence model. And for every nucleotide, it predicts, is this a splice donor, acceptor, or neither? So it basically tries to reconstruct the intron-exon pattern of the gene purely from sequence. And if you go into the model architecture, we actually train a few different flavors of this at 80, 400, 2000, and 10,000 nucleotides of sequence context as input for each output. And you can see that basically these are all residual blocks. This is a ResNet. And I think what distinguishes it from your standard ResNet is that for image recognition is that this is in 1D. And the dilated architecture is, dilated convolution architecture is reminiscent of WaveNet. This is from DeepMind in 2016, but basically it's a way of making your convolutions expand out with every layer so that you're essentially able to cover a large amount of context in a relatively short number of convolutions without doing things like max pooling. And that's critical if you're doing sequence-to-sequence prediction because max pooling completely screws that one-to-one relationship up. Just wanted to pause for any questions.
This is great. Thank you.
Okay, great.
If you have questions, put them in the chat and we'll be answering them as they come.
Yeah, I'll try to blitz through this so that we have at least like 5 minutes to just ask questions and stuff. So this just shows how difficult the problem is. This is the CFTR gene. It's 188 kb for the gene body. And you can see in black, these are the exons. And we asked MaxSense-Scan, which is sort of the previous best PWM-based model. Really nothing wrong with it, to identify where the splice junctions were out of these 188 kb, and it got about 18% of them correct. So red and green is where it marked its guesses. And this is really not bad, given that the gene body is like 188 kb, and splice motifs are incredibly degenerate. But our deep neural network actually predicts all 30 of these exons with nucleotide level precision. And the key is that it turns out beyond the splice motif itself, long-range sequence determinants are key to splice regulation. So what are some of these things? Some of them are like nucleosome signaling. So although we never trained our network to do nucleosomes, we discovered that it also conveniently learned nucleosome positioning because it's apparently quite important for determining exon definition. And then the second thing that we find is that if you look at any genome browser plots of genes, you'll find that exons curiously cluster together. So it turns out that that's not by chance. It appears that when you're in clusters of exons, the threshold for splicing is much lower than when you're trying to break a very long intron. So generally, when you have a splice junction that interrupts the end of a long intron, it needs to be near perfect in its motif. But when you have a bunch of these clustered exons, you actually accept extremely degenerate motifs. So this is like another key insight that the network was able to automatically derive from sequence. And you can see just how the accuracy of our networks goes up as you increase the sequence context size. Yeah, and I just want to show that it also works just on link RNAs too, to just show that we're not overfitting purely on protein coding sequence. So the features that it uses are really no surprise. This is the branch point. This is the polypyrimidine track. This is the AG and the GT, the essential donor and splice dinucleotides. And this is sort of the extended splice site around it. And these are actually intronic splice enhancers, and this is a exonic splice enhancer. So basically, everything that has been reported in the literature, we went and checked to see if the model was actually learning it, and it virtually picked up everything that has been documented over like the past 4 decades of research into the splice determinants. And here's just shown again the impacts of intron length and exon length. So what I've shown is in blue, locomotive score. And what I mean by locomotive score is we created a splice site network that only takes in like 80 nucleotides. And then this is the full model 10K nucleotide score. And you can see that for exons that are sort of average in length, and this is the histogram of all exon lengths, so it peaks at around 140. So for the average exon, you can see that the local network that sort of only looks at the local features thinks that these are actually pretty poor splice sites. But our 10 kb network with the long context actually thinks that the ones in the middle are sort of most optimal. So essentially, this exon length is one of the features that's used by the long range model is one of the things that makes makes splicing more favorable. And as a result, this long range context compensates for the local motif, and it allows the local motif to be more redundant, and therefore it scores more poorly on the local motif score. And this is essentially why the previous generation PWM-based models were completely unsuccessful at predicting splicing at scale. All right, I talked about nucleosome story as well, and let's get to clinical data. So now we applied this to rare disease patients. So this is a patient with early onset heart failure we got from Genomics England, and what we do is we take in the whole sequence of this gene, MYBPC3, and then we flip in the single nucleotide mutation, and then we predict with Splice AI where the exon-intron junctions are. And you can see that here in the mutant, it causes the exon to be extended 14 nucleotides, which frame shifts the protein. So really, if you're just off by even a single nucleotide, the results are usually catastrophic. And in this case, this person has an early onset heart failure before the age of 40 in cardiomyopathy. And then we also took this model to validate on RNA-seq from GTEx. So what we did here is we looked at these novel junctions that were created. So these are, for example, like new exons that were created out of nowhere in the GTEx data, never present only in one person. And what we look for is a singleton mutation only in that person, which also had a high Splice AI score. You can see that there's a huge odds ratio for those events near the ends of these new exons that were created in the RNA-seq data. And this is sort of our overall validation rate. And you can see our validation rate on RNA-seq depends on our Splice AI score. But when you get to high scores, it essentially is pretty similar to the validation rate for essential AG or GT gain losses. And you can see just the validation rate versus specificity for us versus the previous generation PWM methods. And I think one thing that makes interpretation of these cryptic splice variants very complicated is that not only do sort of the weaker variants by our model validate less often, but they're also much weaker in terms of the fraction of transcripts that they sort of bring into aberrant splice forms. So in general, what we find is that for many of these lower scoring splice variants, they actually preserve a lot of normal splicing. So in this case, we find that about 9% of the transcripts have now been changed into creating this novel splice junction that skips 35 base pairs and therefore frame shifts the protein, whereas about 91% still use a normal splice junction. Now, these may be slight underestimates because of things like non-sense-mediated decay. So this could be potentially as much as 30 to 50%. But I think this also adds into the complexity of interpreting these variants compared with sort of the standard model of, oh, is it pathogenic or benign? Now we have a much more graded interpretation of human variation, which I think is something that the field needs to move to anyways and accept. We further look at the impact of natural selection on variants that we predict to have cryptic splice function. So the way we do this is that we look at the exact data set of 120,000 exomes that have been sequenced. And we look at variants that are either common, greater than 1.1% allele frequency, or singleton. And what we see is that there's a very large odds ratio in terms of depleting common variants that have high Splice AI scores. So you can see that this ratio is about 10 to 1, singletons to common variants, in terms of variants which don't have any splice function. But when we look at variants that have predicted high Splice AI scores, this should also be about 10 to 1, but this is actually depleted from maybe 1,000 to about 200. And this depletion is actually very similar to that for protein truncating mutations. So essentially, natural selection is showing us that these cryptic splice mutations predicted by Splice AI confidently are essentially equivalent to a nonsense or frame shift protein coding mutation. And you can see that overall, this is a number of protein truncating mutations per individual, about 11, and there's about 4 of them that we can add on in addition to this that are due to cryptic splice mutations. So it increases it by about 30 or 40%. And then finally, we took this to large clinical data sets. And what we did was we looked at the trio sequencing data for autism spectrum disorder patients and intellectual disability patients. These are each cohorts of about 4,000 trios, plus additional sibling controls. And we looked at de novo mutations, and we predicted whether or not they'd have cryptic splice function based on splice EI. And we find that de novos with cryptic splice function are strongly enriched in cases versus healthy sibling controls. And then we actually went and we took blood from these patients, RNA-seqed them, and demonstrated that we picked up exactly the aberrant splice junction that we predicted using our algorithm. So these are just on top, this is the patient. On bottom, this is a healthy control. And you can see just these variants causing splicing to occur at the wrong location. So here, it moves the exon over. So the exon starts too early as opposed to here. And here's a case where it just completely skips this exon. You can see that in here, there's the vast majority of reads. At least remember these are heterozygous. So it's like essentially all of them on this heterozygous haplotype all go and skip this exon versus this is where normal splicing occurs. There's also lots of examples of novel junctions. So this is a additional intron that's created in the middle of an exon by this variant, and this is a healthy control. And this is another case where the exon starts too early, again causing a frame shift. Anyways, you have a lot of these examples. A bunch of these are in disease genes. This is a case where the intron is fully retained. So it just creates a super exon instead of having normal splicing. And this is a clinical case where this patient had cancer and in multiple organs. And this is typically caused by mutations in MEN1. But when they sequenced the patient, they couldn't find any protein coding mutations. It turns out that the pathogenic mutation is in a non-coding sequence. It's totally non-conserved. No one would guess that this does anything. But actually, SpliceAI is completely confident that this frame shifts the protein by causing the exon to occur too early. So all of our tools are available open source. So please go ahead and play with them. And just wanted to thank my team that worked on this. And if you guys have any questions or you're interested in research positions, internships or postdocs, just send me an email. Let me know if you have any questions.
Thank you so much, Kyle, for a wonderful presentation. Again, we're very fortunate to be able to interact with you. So I think students who are interested in splicing and in this more general space of variant interpretation would benefit greatly from interacting with you. So we'll be in touch as projects take us that way.
Fantastic. Thank you for the invitation. Yeah.
Thank you very much for doing this. And thanks everybody for staying over. Sorry for all the technical difficulties and challenges here. Again, we're so privileged and fortunate to have 3 truly outstanding speakers today. So thank you so much for sticking around. And thanks, Kyle. Thanks to both the previous speakers. And I look forward to seeing you guys again soon and hopefully interacting with you on the final projects. Bye, everyone.
Bye.
Bye.
Thank you, everybody.
Thank you.